The last thing we're going to cover in the citric acid cycle is regulation of the TCA. Here's a table of standard free energy changes in the citric acid cycle for each of the reactions. Just by looking at this table, you can see that only 3 reactions, reaction 1 catalyzed by citrate synthase, reaction 3 catalyzed by isocitrate dehydrogenase, and reaction 4 catalyzed by alpha-ketoglutarate dehydrogenase, are the 3 highly exergonic reactions. What does that mean? If you have a reaction that's highly exergonic, it means it's unidirectional, and thus it is a control reaction as well. And of course, we also know that the first reaction in the pathway is controlled or highly regulated, and the citrate synthase reaction thus is controlled, and that's a given. But which other reactions are controlled? The isocitrate DH reaction and the alpha-ketoglutarate DH reaction are the other 2 controlled reactions as well. Okay, so here if you look at the diagram of the TCA, which reactions have a one-directional arrow, which are not reversible, like this one's reversible, that's reversible, which ones are not reversible, and they would be the ones that are highly exergonic and are thus controlled. And we know that, we've seen that in the previous slide. It's 1, 3, and 4, and here you see that. Here's 1 citrate synthase, that has a one-directional arrow. It's the first reaction of the cycle. It's exergonic. That's what the one-directional arrow means. It's highly controlled. Here's reaction 3, the isocitrate DH reaction. It's unidirectional. And here's reaction 4, the alpha-ketoglutarate DH reaction. It's unidirectional as well. So these are the reactions in the TCA that are regulated or controlled. And one more thing, of course you control a gateway. So the pyruvate DH reaction, the gateway between pyruvate from glycolysis and acetyl-CoA from the acetyl-CoA, the feedstock into the TCA, is also highly controlled. So these are the 4 main control points of the TCA if you also consider the PDH or the pyruvate dehydrogenase reaction as well. And as you can see, that reaction, the PDH reaction, the decarboxylation. Reaction 3, the isocitrate DH reactions of decarboxylation. Reaction 4, the alpha-ketoglutarate DH reactions of decarboxylation. Those reactions normally are unidirectional and are going to be exergonic. Okay, so if you look at the TCA, how is it regulated? The TCA is regulated by 3 simple mechanisms. 1, substrate availability. That makes sense. 2, product inhibition. And 3, competitive feedback inhibition. We'll go over this more when we actually get back to classes after the break, but one thing I want to point out is that all the main regulatory enzymes, the enzymes here, PDH, citrate synthase, isocitrate DH, and alpha-ketoglutarate DH, all of those, the 4 main control enzymes, are all allosterically inhibited by NADH. So what is one of the main products of the TCA? One of the main products is NADH, right? The high energy cofactor that can be used to make ATP in oxidative phosphorylation. So it's one of the main products of the TCA. And if you have the TCA going and you have a lot of NADH present, or if you have a lot of NADH present, you don't need the TCA going, and NADH comes back to allosterically inhibit pyruvate DH, citrate synthase, isocitrate DH, and alpha-ketoglutarate DH. That's a very important point. And one more thing I want to point out is that let me see if I can find it here. Let's actually look at it on the next slide. So let's look at it right here. Okay, here are the main points of the TCA and the PDH reaction. Okay, first of all, it occurs in the mitochondria except for one enzyme, succinate dehydrogenase, and that is the only membrane-bound enzyme of the TCA. Everything else occurs, all the other enzymes of the TCA occur in the matrix of the mitochondria. The next thing is that the reactions of PDH, citrate synthase, isocitrate DH, and alpha-ketoglutarate DH are all irreversible. They're one-directional arrows, they're exergonic or highly exergonic, and they're irreversible reactions. And thus, they also are the main control reactions, right? So all the reactions that are irreversible are also controlled. But what are which are the main control reactions? The main control reactions are the PDH reaction, the gateway reaction, and the isocitrate DH reaction. So if you look at this, all of these, the PDH reaction, the citrate synthase reaction, the isocitrate dehydrogenase reaction, and the alpha-ketoglutarate DH reaction, they're all irreversible, they're all regulated, but the PDH reaction and reaction 3 are highly regulated, and they have the capital R compared to a little r for citrate synthase and alpha-ketoglutarate DH. Alright, what else? The PDH, pyruvate dehydrogenase, and isocitrate DH, the main control reactions, are activated by ADP. That makes sense, right? If you don't have much ATP, you have a lot of ADP, so you want to get ATP made, so you activate the TCA so you can get energy to make ATP. And one more thing to keep in mind. This is what I was going to cover in the previous slide, but it's covered here. Succinyl-CoA synthetase, or succinyl-CoA actually, excuse me, the product right here, succinyl-CoA comes back and is a feedback inhibitor of alpha-ketoglutarate dehydrogenase. So it comes back and inhibits the enzyme that precedes it, alpha-ketoglutarate DH. And this is an example of feedback or product inhibition. One more thing is citrate inhibits citrate synthase. So citrate comes back and inhibits citrate synthase. Another example of feedback or product inhibition. PDH, citrate synthase, isocitrate DH, and alpha-ketoglutarate DH are all inhibited by NADH. That's something we covered in the previous slide. They're inhibited by one of the main products of the TCA, NADH, and that's why you put an N here, 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 and here. So all those main 4 control enzymes are also inhibited by NADH. Finally, the main enzyme that makes the cycle unidirectional is alpha-ketoglutarate DH, and that gets a U there. That says it's making it unidirectional. And then, where do you get substrate level phosphorylation? That's at succinyl-CoA synthetase. You put an S there to indicate that, and it's GTP made by substrate level phosphorylation in eukaryotes, or ATP made. One more thing to point out is that succinate dehydrogenase is also competitively inhibited by malonate because it resembles the substrate. So, this diagram is a really good summary of many of the important points of the TCA. It's kind of chock full of information. Please make sure you know it for exam 4. And you'll need to know some things about regulation for the TCA structure quiz as well. And I like the way these acronyms work. For PDH, it's IRAN, and you know what each of these letters mean, so that'll help you remember that. So IRAN here with all caps. For citrate synthase, it's IRAN. For isocitrate dehydrogenase, it's IRAN again. And for alpha-ketoglutarate dehydrogenase, it's IRAN. And that'll help you remember it, I hope. I think the acronyms work well to help you remember them. Remember that succinate dehydrogenase is the only membrane-bound enzyme of the TCA. It's really also complex 2 of the electron transport chain. And remember that succinyl-CoA synthetase is the only enzyme that catalyzes a substrate-level phosphorylation in the TCA. So again, this is a very important diagram. This is about where I'm going to stop, just to show you a pretty picture of the TCA right here with the different enzymes and showing you their structures from different organisms. And finally, here is an animated review of the TCA. So if you go over this, you can watch this if you watch the PowerPoint presentation and see if you can figure out what's happening in each step. That'll kind of make sure that you know the TCA for the structure quiz that's coming up after the break. I'm stopping here. I hope these notes were helpful. This video presentation was helpful. It's not perfect, but I think it did a decent job of explaining the TCA to you. And I hope you guys have a blessed Thanksgiving, and I'll see you after the break.